Good morning. Good afternoon. Good something. Good day. Good morning. In some time zone. Good morning in Hawaii. Good morning. Aloha. All right. So, finishing up meiosis and mitosis in the very near future. Got a few more slides to cover. I think at this point we've gone over most of the vocabulary items for this topic. We talked about ploidy, cell cycle, the importance of metaphase in viewing chromosomes, what they look like in metaphase. That's how we normally look at chromosomes. We'll see an exception of that in just a minute. The difference between chromosomes and sister chromatids, which we'll talk more about a little bit today, and focusing in particular today on polar bodies. I mentioned that for the first time in the last class. This time we're going to focus a little bit more on polar bodies and analyzing the DNA content of polar bodies and why that might be relevant. I put this back in. We've seen this before, but I feel like the first time I went over it was right before the exam, and so we didn't go fully through the answers to these questions. I want to make sure that we had an opportunity for anybody to ask additional questions on these two views of the same species karyogram. Both human. Any questions about any of this? How many double helices are in each image? What stage of the cell cycle was each cell in? And what are the ploidies of the nuclei? I think the one thing we talked about already about this image was how many double helices are in each image, but we didn't talk about the other two. Yeah? Okay. That's going to make class go really fast. Sure? Yeah.
Whether the two is an early or late cycle of metaphase or part of metaphase.
Right. So the difference between the two in sum is you can see, let's pick a chromosome. Two large metacentric chromosomes, each of those X's really a pair of sister chromatids. Two double helices next to each other or crossing that make that X-shaped thing or pair of X-shaped things. So those are two sister chromatids. And this is in, as you said, metaphase. And the whole point of doing this, again, was to realize that what's on the left here, the picture on the left, the NHGRI karyogram, is exactly the same thing as we see here. These are both human cells. The only difference is that the one on the left is a little bit earlier in metaphase before the sister chromatids have started to separate from each other. So those two things that are circled on the right are the same objects there, for example. So early metaphase on the left, late metaphase on the right. So how many different double helices are in the circled, whatever's in that circle in the upper left? How many total double helices are in there? 4. Because each of those, let's see, choose a different color. That object circled in red, either one of those is the same thing as one of those on the right. So it's either looks like a stick, two double helices next to each other, side by side, so close together you can't really distinguish the separation of the sister chromatids. Two double helices in each item up here that's circled in red. So the two pictures have the same number of objects, what we consider chromosomes, 46 on the right here, 46 things on the left, just different stages during the cell cycle. And each of these are diploids, so there are two copies of each object. That's 2n equals 46. So that's the three questions. How many double helices are in each image? If the total number of objects is 46, then the total number of double helices is, if this thing here, this X-shaped thing, is one of the 46 objects, and it's 2 double helices, then the total number of double helices is twice 46, which is 92. So 92 double helices in each image. Metaphase, both metaphase chromosome spreads, both are diploid. 2 copies of every chromosome. 2 homologs in the terminology that we've used before. Anything else up here you see that's odd, interesting, different, you want to ask questions about? Okay. So here is the ultimate picture. This is my favorite picture of a karyogram. This is not on the, well, this might be on the test. The fact that it's my favorite karyogram will not be on the test. So what's the ploidy number of this metaphase chromosome spread? So if it's diploid, we don't know yet, what do we have to do to know? Sure. So if we need to be able to arrange them like a karyogram, pair them up, and see if there are pairs or triplets. What if it was triploid? There would be 3 chromosomes up there that look the same, 3 pairs of sister chromatids. What do you think? Are there 2 pairs of 3 or 3 pairs of 2? Which chromosomes look the most alike? If I call the one on the top right number 1, I'll just number them all. You tell me which ones you think are the most similar to each other in shape and size.
4 and 6.
So I heard 4 and 6. I heard
1 and 5.
1 and 5. So that would leave 2 and 3. Right. So those chromosomes look similar in size and shape. So this is a diploid, 2n, 2 copies of every chromosome equals 6. 6 X-shaped things up there. 6 pairs of sister chromatids, there are 6 chromosomes. It's a diploid. And the reason that I like this chromosome spread in particular is because it's from the mammal that has the fewest number of chromosomes, the muntjac. It's an Asian hoofed mammal. They've only got 3 pairs of chromosomes, which is interesting to cytogeneticists just because you can actually see all the chromosomes in a tiny little space. On the right side, what are we looking at? Well, I've labeled it, but can you explain what we're looking at? What are the different colors, do you think?
Different chromosomes?
The different chromosomes have been artificially colored. They've been dyed different colors. So you can see, let's see, metaphase, they don't look like X-shaped things. All of these sort of fluffy pipe cleaner looking objects down here in the bottom part of the picture. So what stage in metaphase is that going to be?
Early.
This is early, before the sister chromatids have spread apart. This is really early metaphase. Both of the sister chromatids are right next to each other. We can't see any separation between them. And the reason I like this picture is because it underscores the point about chromosome shape and only being able to see it at metaphase. Because what does the interphase nucleus look like in the top? This is one entire nucleus that the scientists did exactly the same process to. They colored all the chromosomes, but during interphase, what do the chromosomes look like? They're in a 3-dimensional sphere. They're in the nucleus. They're decompressed. They're unwound. So they fill completely the nucleus, and each chromosome is in, this is the really weird part. This sort of thing freaked scientists out the first time they saw it. They didn't know what chromosomes did in the nucleus during interphase in terms of where in the nucleus each chromosome would be. I mean, it could be that every chromosome unwinds and fills part of the nucleus, and that every other chromosome also unwinds and fills the whole nucleus, in which case you'd see green and purple and red throughout the whole nucleus. It would look like one color if every chromosome was everywhere. For example, you could imagine that one chromosome would unwind like that, and another chromosome would unwind like that, and a third one, and they would be all mixed together in the nucleus, but that's not what happens. This picture shows that when the chromosomes decompress during interphase, they don't just go everywhere, they stay in one region of the nucleus, which was really surprising to scientists. They just said they never knew that before. This shows that chromosomes actually live in specific regions of the nucleus during interphase. Any questions about that? Human chromosomes do the same thing. When our chromosomes are in interphase, they live in particular, they reside in, they take up certain areas of the nucleus. They're stereotypical.
So I noticed that the two blue, I think those are blue, chromosomes have a little section of a different color, red.
Mm-hmm.
Does that mean that they've crossed over, or is that just how it is, or?
That's just how it is. That's the easy answer for me, right? Whew, I'm done. The way that they colored these chromosomes is a particular technique that uses chromosomes themselves, colored chromosomes, as the dyes. So what you might do, this is something we'll talk about later in the class, not today, but later in the semester. You could extract one of these chromosomes and fluorescently label all of the helix, all the double helix of this chromosome, and then you could melt all of the chromosomes, you denature them all so they're single-stranded, you throw in this fluorescently labeled chromosome, and then it will base pair to the chromosome that it's complementary with, and that's how they get these fluorescent images with every chromosome stained a different color. So the fact that there's red on multiple chromosomes suggests there's some sequence on the red chromosome that's also present on the blue chromosomes and the green chromosomes, or whatever colors those are. My apologies if I'm not getting the right color terminology. Chartreuse and magenta. Yeah.
Does that not show like the centromere?
It's hard to see where the...
Is that what the red color is?
So it could be the centromere. And I actually was thinking that myself before class. I was trying to figure out what those red colors were, and it's very likely that those red parts are the centromeres. I'd have to go back and look at this particular paper. But if you compare, and you can do this on your own, we can do it right now, I'll leave it as a thought question. If you look at the centromere positions of the chromosomes on the left, do those match with the red dots in the chromosomes that are over here on the right? Does it look like the centromeres might be where these red positions are on the different colored chromosomes? Obviously, you can't tell on the red chromosomes because they're already stained red. But on the other two, might it be that those are the positions of the centromeres? Hey, cruising right through. There's no reason to cruise through, by the way, so please ask questions anytime you have any. One of the papers that I'd asked you to read this semester so far, and the one that I had asked you to look at for today, genome analysis of single human oocytes. So I want to address this question as a group, and then I want to give everybody time to look at the next slide, the one figure that I asked you to look at for today. And maybe in a small group or by yourself or with a partner, go through those questions, make sure that you're on the same page with people around you, and then we'll talk about the answers to those questions. But first, I want to make sure everyone's all together on the same page before we go off on that little adventure. What are these scientists trying to do? If you looked at this paper, what's the problem they're trying to solve? There's always a question, a hypothesis. This one's actually a very applied problem that these authors are trying to solve. A study they did with human oocytes. Anybody pick up on what their goal was? Well, let's do a quick review of meiosis. Again, another quick review of meiosis. How do human male and female meioses differ? They have the same number of divisions. Let me change color here. Start with a diploid cell. Then what? Meiosis is going to start. The first thing that happens is chromosomes have to Segregate, replicate, replicate. Synthesis. Temporarily have 4 copies of every chromosome, 4C, technically, not 4N, but N is fine. So on the right side, we've got male. Let's say on the left side, female meiosis. How many cell divisions? 2. So in the first cell division, what does this look like? Division 1, to the left, males, to the right, females. What does it look like in males when the first cell division happens? What are the resulting cells? Say it loud, say it proud. What was it? You get 2 cells, right? One cell divides into 2. Each cell has how many copies of each chromosome? Right, 2. There's 4 copies, they have to divide some. It's either 1 and 3, 0 and 4, or 2 and 2. 2 and 2 is the ideal situation. How does this look different in females? Do you still get 2 daughter cells?
You have one that's really big and one that's really small.
You still have 2 daughter cells, but you get one that's really big, one that's bigger, and one that's smaller, and that's called a polar body. So how many chromosomes are in each of those 2 cells? Is it the same as males, or is it different than males? Same. They still divide, in theory, hopefully, they still divide equally. 2N going to each of those daughter cells, and then during the second cell division, each of the male cells divides equally and produces 4N, single copy of every chromosome gametes. The female, the big cell, divides and produces, is this one an equal or an unequal division in terms of volume of the cells?
Unequal.
This is also unequal, produces the N, the oocyte, the functional gamete, and then another polar body, the second polar body. And the first polar body can divide again and produce 2 more N.
And those are technically also polar bodies.
Yeah, those are also polar bodies. They're the daughter cells from the first polar body. So that's DNA this is really important for this paper. That's the DNA content in all of these cells, especially in females on the right, through the process of meiosis. So what's the point of this paper all about? Genome analyses of single human oocytes. Do you know how in vitro fertilization works? Kind of, sort of, you have an idea? What do you think? How do you imagine, if you've never done it before, if you haven't been involved with it, I'm assuming many of you haven't, but I don't know. What do you think happens? In vitro fertilization, test tube babies. What's the doctor gonna do? Okay, so you take an oocyte from a woman, throw some sperm on, one of the sperm fertilizes the oocyte, and then normally you implant that embryo into a surrogate mother or something. Right? That's not genetics. That's developmental biology. That's biology 155. Take it every spring, or at least every other spring. I highly encourage it. It's a good class. I don't teach it anymore. That's not why it's a good class. But it is a good class. So if you're doing in vitro fertilization, we've been talking so far about aneuploidy, the probability, especially at the end of last class. I talked about a couple of manuscripts that have been talking about how early in life gametes are more likely to be aneuploid, have the wrong number of chromosomes, and then there's some peak at which you're most likely to produce gametes that just have 1 copy of every chromosome, which is ideal. And then as you age further, there's a higher probability of having gametes that have the wrong number of chromosomes. So if you're doing, especially if you're doing in vitro fertilization, wouldn't it be nice to be able to know whether or not the oocyte that you're going to use to produce another human had the right number of chromosomes? Seem like a good idea?
Maybe.
Maybe. Well, who's to say? It depends maybe on your religious leanings or your personal beliefs, but sure, if you're going to be a person that's already thinking about doing in vitro fertilization, or you want to do in vitro fertilization, if you're already paying the money to do it, then yeah, maybe it's worth the investment to try to figure out, of these 50 oocytes that have been collected from a woman, which one is the best? Gotta be careful here by what we mean by best, because we don't want to get necessarily into eugenics, choosing what are the best traits, but maybe we could all, maybe, I don't know, maybe we can all agree that a beneficial trait for any human would be to have 2 of every chromosome. And that we'd want to choose an oocyte, therefore, that has 1 and only 1 of every chromosome. That's what this paper is about, is trying to figure out how could we figure that out in human oocytes. Is it possible, especially in the case of in vitro fertilization, there are other reasons you might want to be able to do this, is it possible to tell which of many oocytes are euploid? 1 copy of every chromosome. So how would you do that? If you were a scientist and you had the power of all the scientists in the world and you had infinite resources for laboratory equipment, what do you think you would do to tell which of many oocytes has the right number of chromosomes? Yeah.
Isn't there a way to stain a cell to see the density of how much DNA is in that area?
Okay, so you might want to do something that has to do with measuring the amount of DNA in a cell. So one way might be staining it somehow. What's another way you could tell how much DNA is in an oocyte? You can use your imaginations if you want to. You don't have to know. What are other ways to figure out what DNA content is inside a cell?
Look at it with a special crazy microscope
Sure.
so I can see how much DNA is in there.
Yeah. So you could use, we've done a lot of microscopy to look at chromosomes so far in class, so that's a good answer. Or you could take the chromosomes out of you could rupture the cell, you could take the chromosomes out, you could weigh them. We know how much the mass of a nucleotide is. So if you had a really, really crazy, super fine scale balance, you could weigh the chromosomes, or weigh the cell and figure out how many chromosomes are in the cell. There are a lot of ways you could do it. But what's the drawback with all of these approaches? Right. If you do anything to measure the amount of DNA in a cell, you're doing something with the DNA in the cell. You've put a dye in the cell. You've taken the chromosomes out of the oocyte. Right? None of these things are useful for making a baby because once you've figured out, oh, that was the right egg. On to the next. No, can't do that either. So that's what this paper is about, is trying to figure out, can we figure out how many chromosomes are inside the egg without messing with the egg? We need to find a way to deduce, to calculate, to predict how many chromosomes are in the oocyte. So let's see how this goes. Take a couple minutes. Hopefully you've read it or at least looked at this paper. Have a quick discussion with a neighbor or with yourself. Look at the paper the first time if you haven't yet. Just try to figure out these questions. What are the axes on these graphs? What's the y-axis value? What's the x-axis? What are these data that are plotted here? What do they mean by predicted and confirmed here? And what did they find that was not expected? 2 minutes maybe. Have some conversation. I'll come around and answer any questions you might have, and then we'll come back together and discuss what this means and how they did this. What's maybe the easiest question to answer? If there is one easiest question that I've written up here, what would be the easiest one? Okay, what are the axes? So what's on the x-axis? Right, it's human, and what gives you a hint that it's the number of chromosomes? Yeah, there's 22, and then there's the sex chromosome, an X. Okay, so that's just chromosome 1 on the bottom starting at nucleotide 1, the left end of the chromosome nucleotide sequence. So the other unsurprising thing here is that what do you see about the lengths of the different bars that represent the different chromosomes?
They get smaller.
The biggest one's on the left, that's chromosome 1, because we know, at least we should know by now, I think, that the human chromosomes are numbered in order of size. So they get smaller as they go to the right, and then there's the X chromosome, which is relatively large. What's on the y-axis? Of each of these, there are five different plots. 1, 2, 3, 4, 5. They're all the same type, so once we figure this out, it should be a pretty easy go to understand what's going on here.
Number of copies.
Yeah, copies of chromosome.
Right, so it's ploidy, essentially. So, four would be tetraploid, or 4 copies of every chromosome anyway. What are the different cells? Those are the different panels. What are they looking at here? What components of meiosis are they studying in each of these different graphs? So this top panel has 2 copies of chromosome 1 is what that data point says. What is the thing that they're looking at that has 2 copies of chromosome 1?
The first polar body produced by.
First polar body. So remember back to meiosis from 5 minutes ago. First cell division produces which polar body? First polar body. First cell division produces the first polar body.
And it should be 2N.
And both of the products, the oocyte and the polar body, that's the primary oocyte, should both be 2N. This is, by the way, in case you're starting to zone out now, which is totally possible because it's after lunch and this is genetics, all I'm going to ask you to do is addition and subtraction. So hopefully we're up to the challenge here. Because the math is really simple. If you start out with a cell that starts meiosis that has 4 copies of every chromosome, then there are only 4 copies of chromosome to go around in all of the subsequent cells. So if that first polar body here has 2 copies of chromosome 1, then how many copies of chromosome 1 are left to be distributed into the rest of the cells? Started with 4N. We've got 2 in the primary oocyte, or the primary polar body. Started with 4. We've accounted for 2. So there are 2 left. That's the subtraction part. So where did they go? In the second round of meiosis, we get the second polar body. It should be 1C, 1 copy of every chromosome. And is that what it has?
No.
For chromosome 1, the whole length of that chromosome, it's got 1. And so what does that mean is left? We've looked in the primary polar body. The first polar body has 2 copies of chromosome 1. The secondary polar body has 1 copy of chromosome 1. So what's in the oocyte? The last chromosome that's available to be segregated into the oocyte. We've already accounted for 3 of the 4 by looking at, this is the trick, they don't actually destroy the oocyte to figure out what chromosomes it has. They take the primary polar body and the secondary polar body, they break them open, they count how many chromosomes are in those cells, and they subtract the total in those cells from 4, and that's how many chromosomes are in the oocyte. So, 4N, you get the first division, you get the second division. So if you destroy these cells, the polar bodies, well, technically that's not true. What they've done is they've taken the first polar body after the first division, and the second 2 polar bodies, the second polar body rather, at the second division. And they've sequenced those, basically. So first polar body goes there, second polar body goes there. So the first polar body should always have 2 copies of every chromosome. The second polar body should always have one copy of every chromosome. And what did they find? Was that true for all the chromosomes in this oocyte that they're looking at the polar bodies of? Is there any place where there is not two copies of a chromosome in the first polar body and one in the second polar body?
In between the dashed lines?
Okay, so the authors have helped us out here a little bit. They put a bunch of dashed lines in here to highlight specific parts of specific chromosomes. They're trying to highlight that there are, in this one round of meiosis, first cell division, second cell division, produces one oocyte. They collect the polar bodies, they take the chromosomes out of the polar bodies, they measure how many copies of each chromosome. There are a few different regions of the genome where what happens in the oocyte. So let's focus on this bit right here, chromosome 16, that little bar.
The female pronucleus has none.
If there's 3 copies in the first polar body, that's this one, that's 3N for chromosome 16, then how many chromosomes are left to go between either the oocyte or the second polar body?
One.
There's 1 left. In this case, where did that 1 chromosome go? Did it go in the polar body or the oocyte? The polar body's got 1, so they predicted, just by doing that math, destroy the 2 polar bodies, find out how many copies of chromosome 16 there are in them. They do the math and they predict that if they looked in the oocyte, they would see 0 copies of that chromosome. So in this study, because they weren't actually interested in doing in vitro fertilization, they just wanted to prove the point that you could use this for genetic testing to do in vitro fertilization, they did actually destroy the pronucleus, the oocyte, to actually measure experimentally how many copies of every chromosome, and it was 1 until they got to chromosome 16, and that oocyte did indeed not have a chromosome 16. So would you use that oocyte for in vitro fertilization? Up to you, but probably not. What else did they find besides this oocyte lacked a copy of chromosome 16? So we're looking for anywhere where the female pronucleus predicted or confirmed, either one, the pronucleus, the oocyte, where does it deviate from having 1 of every chromosome? It's missing a chromosome 16. It's got an extra chromosome 19.
No.
It's got no chromosome 22. Right? This is not a great oocyte for having a kid with, probably. Plus, what have I missed?
Part of chromosome 2 is a little bit wonky because the first polar body has 3, the second polar body has none, but the pronucleus has 1.
Interesting. And this is just part of this chromosome, the left end of chromosome 2. Not all of, because all of chromosome 2 is all colored in blue here. So it's not the whole chromosome 2, it looks like part of chromosome 2 has broken, and there's an extra bit of it in one cell, so there's not enough of that part of that chromosome in the other cells, but thankfully the pronucleus was fine. So those bits of random chromosomes happened to be put into the polar bodies, not into the oocyte, in this case. That's not necessarily always going to be true, but it happened this time. So sure, chromosomes can break during cell division too, and then the bits of extra or not enough chromosomes segregate however they will. They go into different cells.
I don't know if you know this, but was the data taken from multiple different people, or was this all one person with all of their gametes?
This is one meiotic event from one person.
Okay.
This figure is all from following one tetraploid, one 4C cell, one cell with 4 copies of every chromosome watching it go through meiosis.
Okay.
But they do this a number of times to know if this is a repeatable approach that you could theoretically use. But this graph is just from a single oocyte and its 2 associated polar bodies. Any questions about that? So it's the simple math. Start with 4 copies of every chromosome. They've got to go into a limited number of cells. If you know all the content of all the cells but one, you can mathematically predict what will go in the other cell.
Are the polar bodies destroyed within our bodies, or are they sent through with the oocyte?
The polar bodies eventually die, but they're not destroyed in specifically not destroyed. They're just not used. They no longer divide, which is equivalent of death to any cell. If a cell stops dividing, it's still alive. It's probably still doing things, but if it's no longer dividing, it's like, it's going extinct, let's put it that way. It's like if humans stopped reproducing, then we would all go extinct. Does that answer the question? I feel like I dodged a little bit.
I'm just wondering, like, if you wanted to do this in vitro fertilization, and they gave you the hormones to ovulate, and what are they isolating? Are they isolating a 4N cell, or are they going to isolate an oocyte along with polar bodies?
So there's a great figure in this paper that I think it's in this paper that shows the technique. It shows them actually collecting, it's a drawing, it's not a picture. It's of collecting the polar bodies because they sort of adhere to the oocyte. So you know, if you collect an oocyte, you know which polar bodies are its polar bodies because they're all associated with each other physically. All right, in the last, so we've got 12 minutes. Take about 10 minutes and do this last question. Thought experiment with a calculation involved. So again, I'll ask you to work in small groups if you want to. I'll circulate and answer questions if you want to. The big goal here is this is the end of meiosis for now. We're going to move on to studying alleles next class. One question that a lot of people are curious about is why siblings kind of look similar to each other, usually, sometimes not. One of my siblings looks very different than my other sister and I. But in general, why do we look similar but not identical? Well, why don't we look identical? Why don't most siblings look identical to each other?
Because they don't necessarily have the same exact gene inside of them because of the fact that you get like one copy from mom, one from dad, and there's a crossing over and a whole bunch of other stuff.
Yeah, and a whole bunch of other stuff like crossing over we'll get to later in class. But yeah, right, there's genetic variation. Each of your parents can give you certain combinations of chromosomes, and so no two sperm and no two eggs that are used to create you and your siblings or cousins are identical. So the question here is, getting to that specific question. We've seen last class that if you have a cell that's about to divide, you could have, for example, in black, the 4 copies, the 4 sister chromatids of chromosome 1, let's say. 2 paternal, 2 maternal. We have the same thing happening with chromosome 2 in blue, let's say. 2 copies of the paternal chromosome, 2 copies of the maternal chromosome. We talked about the fact that these can go, for example, the 2 copies of chromosome 2 can go in either order. The paternal copies could be on the left side and maternal on the right, or it could be mirrored. It could be the 2 maternal copies on the left side here, and the 2 paternal copies on the right. And we have 23 pairs of chromosomes. And they can all independently decide which side of the cleavage furrow, which cell they're going to go into at the first division in meiosis. So the question here then, we've done this math in class before, so you know the mathematical approach, we just haven't used it in this context yet. How many different combinations of maternal and paternal sister chromatids are possible at meiosis 1? That is, if you were going to line up, another way to think about this is if you were going to line up the 23 sets of sister chromatids that go in the left cell, how many different combinations are there? Maybe the easiest way to think about it is what's the probability, and then I'll let you go, what's the probability by chance that all of the paternal versions of all 23 of the chromosomes all line up on the same side of the cell? That's the question. How many different combinations of chromosome arrangements are there in meiosis 1 for humans? Try that. Please do confer with neighbors if you want to. Ask me questions, and we'll come back together in about 5 or 6 minutes, if necessary. You've got at least that long if you want it. Who thinks they have an answer? Did anybody actually calculate an answer? So, what are your answers? It's
Okay, I'm not gonna say, but it's.
okay.
I did, 1/2 to the 23rd.
So I hear 1/2 to the 23rd. Other approaches? Yeah.
2 to the 23rd?
2 to the 23rd. So the distinction between the two is, are we talking about probability or frequency?
No, because I did 1/2 to the 23rd power, and I got 1.192 times 10 to the -7. Probability.
So that's the probability, which is a really small number. If we take its inverse, 1 over that, that's something like, let me see, I did the calculation, 8.38 million.
And that's how many different.
There are 8.38 million different combinations of chromosomes that can be put into a gamete of meiosis in humans.
For any one person.
From one person. So why is that the quickly. So here's chromosome 1. The paternal version, oh, I should have colored those black. Yep. Paternal and maternal version. What's the probability that, you pick a direction, left or right?
Left.
Okay, left. What's the probability that the maternal version is going into the left cell? It's either going to go in the left cell or the right cell. So there's a 50% chance that it goes into the cell on the left or the cell on the right. So if you look at chromosome 2, sister chromatids at meiosis 1, what's the probability that the maternal versions also go into the left side of the cell? It's also 1 and 2 by themselves, not worrying about any other chromosome. There's always a 50-50 chance that the paternal or the maternal versions will go into one or the other cell. So you just multiply 1/2 times 1/2 times 1/2 times 1/2 23 times. That's why the answer is, you've got a 50-50 chance, and there are 23 pairs of chromosomes. So, right, what's the chance that any one of us, or any human for that matter, doesn't have to be one of us, what's the probability that any human in their lifetime produces 2 gametes, 2 sperm or 2 egg, that are genetically identical? There are 8.38 million different combinations of chromosomes. It's harder to answer for guys. And by the way, if you want to jump into Socrative now, you can. I just opened a quick couple of questions. But to end, how many sperm do guys make during their reproductive lifetimes? I have no idea. But I could Google how many oocytes do women produce. It's a smaller number, so it's maybe easier to think about. So how many oocytes do women produce or have the possibility of using during their lifetime? Let's do a quick and dirty back of the envelope calculation. How many years are women fertile typically?
About 40, I would say. If you get your period at 10, you go through.
Okay, so let's say 40. This is a quick and dirty calculation. Let's say it's 40 years, 12 menses per year.
180.
So that's on the order of hundreds, 200, 400, 300.
Yeah.
There are 8 million different possible combinations of chromosomes. So you don't even touch the total possible combinations of chromosomes that you could produce to make kids. So why are siblings not genetically identical? No chance that they would both get the same sets of chromosomes from both parents that were the same sets of chromosomes from the sperm and the egg. See you next time.